Hello students, welcome to EPG Pathshala. I am Dr. Jaswinder Kaur from Gargi College, University of Delhi. Today we are going to talk on module In-situ Hybridization from paper Methods for Gene Identification. After studying this module, you should be able to learn the basic protocol followed for performing in-situ hybridization, the various probes utilized, and the labeling techniques used. Further, the applications and limitations of the technique will be discussed in brief. In the end, fluorescent in-situ hybridization will also be discussed in brief. The technique was invented by George G. Gall. In-situ hybridization, or ISH, is based on the complementary base pairing of labeled cDNA or RNA probe to normal or abnormal nucleic acid sequences in chromosomes, cells, or tissues, mostly to detect and localize specific mRNA sequences within preserved tissues and cells. Furthermore, morphological identification is possible in a heterogeneous cell population. That is, from such a heterogeneous population, actual affected cell can be identified. The sensitivity of the technique is detection of even 10 to 20 copies of mRNA per cell. The technique is particularly useful in disease pathogenesis and neuroscience, where detection and localization of a particular mRNA sequence is required. This technique is distinct from immunohistochemistry in localizing DNA or RNA within cells. Immunohistochemistry, on the other hand, localizes proteins such as antibodies in tissue sections by antigen-antibody interactions. The basic principle of ISH is same as that of normal hybridization. In regular hybridization, DNA or RNA has to be isolated from cells, run on a gel, transferred on a nitrocellulose or nylon membrane, and then hybridized with complementary sequence of interest. ISH, on the other hand, is used to detect and localize the sequence of interest in situ, in the cells and tissue section itself. However, detecting the sequence in situ presents certain difficulties. The mRNA sequence of interest can be present in minute quantities, it can be masked by some associated proteins, or it might be protected by some cellular structure. So, without compromising the structural integrity of the cell, the permeability of the cell membrane or tissue has to be increased. The sequence of interest should also be made better visible to the probe. For this, choosing the correct type of probe with correct labeling is crucial. The tissue sections to be used can be prepared in different ways. First, in frozen sections. Fresh tissues can be snap frozen by applying -80 degrees temperature. These frozen sections can be embedded in special support medium prior to cryosectioning. The sections are then fixed in 4% paraformaldehyde and used for hybridization. Second, one can also use paraffin embedded sections, in which the sections are first fixed and then embedding is done in wax before sectioning. However, if cells are to be used, then cellular suspension is made. The cells can be fixed onto slide with methanol. General procedure. The process of in-situ hybridization involves 3 basic steps. First, pre-treatment. The tissue and cells are first treated to fix the target transcripts in place and to make them more accessible to the probe. Tissue sections must adhere well to specially treated glass slide to prevent loss of tissue. Adhesives like poly-L-lysine and gelatin chrome alum can be used for this purpose. Various enzymes and detergents are then used to unmask the DNA or mRNA so that they can be bound to the probe. To increase accessibility, dewaxing of the tissue is done in case of embedded sections. Second, hybridization. Heat or alkali treatment is then used to denature the double-stranded probe or target molecule. The single-stranded molecules are then annealed using a hybridization mix to form double-stranded hybrid. The probe with the label now gets hybridized with the target DNA or mRNA sequence. The selected probe is then hybridized to the target at high temperatures. To increase specificity, low salt and high formamide concentration should be used. Post-hybridization washes with RNAs can then be done to hydrolyze the unhybridized probe, which can then be washed away. Increased stringency results in increased specificity and lower background signal. Non-specific reactions can also be reduced by manipulating the reaction parameters like temperature, pH, salt concentration, etc. Third, detection. The labeled probe, whether fluorescent, radio-labeled, or antigen-labeled, can then be localized and quantified using fluorescence microscopy or immunohistochemistry techniques. The detection method depends upon the type of probe and label used. Specific probes can also be synthesized by cloning or by oligonucleotide synthesis. This technique can be used to locate DNA sequences on chromosomes, detect RNA or viral DNA. Probes. Complementary sequence of nucleotide bases to the specific sequence of interest, such as mRNA, are referred to as probes. These can be 20 to 30 base pair to up to 1000 base pair in length. Depending upon the task, different types of probes can be chosen. 4 types of probes are currently available to carry out the process of ISH. First, oligonucleotide probes. Probes can be synthesized synthetically from commercially available oligonucleotides if the target sequence is known. These are small 40 to 50 base pair, so can penetrate the target cell or tissue section easily. They provide the additional advantage of being resistant to RNases and being single-stranded, circumventing any chances of renaturation. Second, single-stranded DNA probes. They are 2000 to 500 base pair in range and are expensive and time-consuming to produce. These can be produced by reverse transcription of RNA template or by PCR amplification. For preparing the probe from PCR amplified product, the product of first round of amplification is used as a template in subsequent round for generating an extended fragment, but using only single antisense primer this time. Thus, single-stranded DNA would be produced. Third, double-stranded DNA probes. If the sequence is known, then appropriate primers can be used to amplify the desired fragment, and a relatively clean preparation of a double-stranded DNA can be obtained. However, if the sequence is not known, then the fragment can be cloned in a plasmid and replicated in a bacteria, lysed to obtain the plasmid, purified, and then restriction enzymes can be used to excise out the fragment of interest to be used as a probe. Fourth, RNA probes or riboprobes. RNA probes are 250 to 1000 base pair in length. RNA-RNA hybrids are very thermostable and are resistant to digestion by RNases. So, post-hybridization washes with RNAs can be done, making it possible to remove any background stain and remove any non-hybridized RNA. The RNA template can be transcribed in 3 to 5' direction using RNA polymerase to make complementary RNA. Labeling techniques. To check where the target gene is being expressed in the cell or tissue section, a reporter molecule is attached to the probe before hybridization. Reporter molecules can be radioactive or non-radioactive. Radioactive labels. Radioactive labeled probes can be localized and detected using autoradiography. The high energy emitted by the radioactive probes is used in reducing ionic silver in the emulsion or X-ray to metallic silver. This signal gives the localization of the probe in the tissue or cell. Radioactive reporters include H3, P32, S35, I125. The most commonly used isotope for ISH is H3 and produces high-resolution autoradiographs. Although it allows precise localization of signals, but long exposure times are needed. However, for low copy number detection of transcripts, S35 is the label of choice. Non-radioactive labels. An oligonucleotide stretch of 5 to 50 modified nucleotides can be attached at the 3' end, known as a tail, using enzyme terminal transferase. These modified nucleotides has a label attached that can be detected after hybridization. There are various labels that can be used. These include biotin, deoxygenin, alkaline phosphatase, rhodamin, and fluorescent dye. More than 2 probes can be visualized at the same time with the use of fluorescent labels. The technique wherein fluorescent label is used is popularly known as FISH or fluorescent in-situ hybridization. Although radioactive labels are more sensitive than non-radioactive labels, but they suffer from other disadvantages like long processing time, poorer stability, and more stringent waste disposal processes. However, the sensitivity of non-radioactive probes is fast catching up with that of radioactive ones. But for the detection of low copy number mRNA, radioactive probes are still the labels of choice. Limitations. In situ hybridization requires good preservations of cells and tissue sections to be studied, such that they can be identified morphologically. The cells and cellular structures should be distinct and intact. Together with preservation, strong hybridization signal is crucial. However, fixation decreases the accessibility of the probe to the target. This happens due to the use of fixatives like formalin, paraformaldehyde. This not only reduces the diffusion of probes into the tissues but also leads to cross-linkage of nucleic acids and proteins. To circumvent this problem, frozen tissues can be used. However, this leads to a compromise of morphological integrity of cells and tissues. The cellular structures and membranes impose hurdles for probe penetration and thus hybridization efficiency. Although a powerful technique, this is an expensive, labor-intensive, and time-consuming process and requires knowledge of bacterial expression, cloning, and in vitro transcription. Applications. ISH technique has various applications in pathology, developmental biology, sex determination, chromosome mapping, and microbiology. Pathology. The technique has majorly been applied in histopathology and detection of viral diseases. It has been applied for the detection of cytomegalovirus, human papillomavirus, Epstein-Barr virus, hepatitis B virus, mycoplasma, salmonella, etc. Typing of infective agents genome in the cells and tissues being studied can be an indicative of clinical diagnosis and outbreak investigations of an epidemic. The actual cells harboring the infection can be localized by ISH, as in the case of HBV infection in hepatocytes. Thus, mode of transmission of viruses from cell to cell can be studied. Second, developmental biology. Cells developed from a progenitor can be identified by the presence of same mRNA. Also, segmentation pattern or body pattern formation in Drosophila can be studied using FISH. Third, sex determination. Sex determination and thus identification of donor and recipient cells in transplants can be done using this technique. Sex is determined by hybridization with a Y chromosome specific repeat. Fourth, chromosome mapping. Genes belonging to same gene families can be detected by hybridization with suitable labeled probes under low stringency conditions. Heterochromatin can also be differentiated from euchromatin by this method. It can be used to detect numerical chromosomal aberrations. Chromosomal deletions and translocations can also be identified. Fifth, microbiology. Labeled 16S rRNA gene can be used for identification of bacterial cell populations. It can be used in the quantification of specific types of bacteria in microbial populations without the need to culture these. Only living cells contain sufficient rRNA to allow detection by fish. A special variant of ISH is fluorescent in-situ hybridization or FISH. FISH is a cytogenetic technique that uses fluorescent probes to detect the presence or absence of specific DNA sequences on chromosome. FISH is vividly used in chromosome painting. In chromosome painting, multiple fluorochromes can be used, which produce a multicolored and painted effect with unique color at each hybridization site. This can be used to identify cross-species homology by labeling probes from one species for hybridization with chromosomes of another species. Other uses of FISH include localizing rRNA targets in cells, detecting tumor cells, gene mapping, ploidy determination, analysis of chromosomal structural aberrations, and patterns of gene expressions in cells and tissues. Telomere probes can also be used to define chromosomal boundaries, that is 5' or 3' ends. So students, let us now summarize what we have learned in this module. ISH or in-situ hybridization is based on the complementary base pairing of labeled cDNA or RNA probe to normal or abnormal nucleic acid sequences in chromosomes, cells, or tissues. It is used mostly to detect and localize specific mRNA sequences within preserved tissues and cells that can be identified morphologically in a heterogeneous cell population. It can also be used to detect and localize the sequence of interest in situ in the cells and tissue section itself. The technique is particularly useful in disease pathogenesis and neuroscience, where detection and localization of a particular mRNA sequence is required. Complementary sequences of nucleotide bases to the specific sequence of interest is known as probes, which are used for detection. Probes can be 20 to 30 base pair to up to 1000 base pairs in length. Depending upon the task, different types of probes can be chosen. 4 types of probes are currently available to carry out the process of in-situ hybridization. Probes can be synthesized synthetically from commercially available oligonucleotides if the target sequence is known. Single-stranded DNA probes are 2000 to 500 base pair in length and are expensive and time-consuming to produce. These can be produced by reverse transcription of RNA template or by PCR amplification. However, if the sequence is not known, then double-stranded DNA probes can be used. The fragment can be cloned in a plasmid, replicated in bacteria. Bacteria then can be lysed to obtain the plasmid, and the plasmid is then purified, and in the end, restriction enzymes can be used to excise out the fragment of interest to be used as a probe. RNA probes, on the other hand, are 250 to 1000 base pairs in length. RNA-RNA hybrids are very thermostable and are resistant to digestion by RNases. So, post-hybridization washes with RNase can be done, making it possible to remove any background stain or non-hybridized RNA. To see where the probe has hybridized to the target DNA, a label or reporter molecule is required. Reporter molecules can be radioactive such as H3, P32, S35 or non-radioactive such as biotin, deoxygenin or alkaline phosphatase. In-situ hybridization is seen as an expensive, labor-intensive and time-consuming process and also requires knowledge of bacterial expression, cloning and in vitro transcription. Still, it is a very powerful technique and can be used in various fields including developmental biology, pathology, sex determination, chromosome mapping, and microbiology. Thank you.